Welcome everybody. So today we've got a fantastic session with 3 speakers, so you're extremely lucky. First of all, we acknowledge and celebrate the first Australians on whose traditional lands we meet, and to pay our respects to the elders of the Ngunnawal people, past and present. So today we're going to hear about rare diseases. So rare diseases is a very special topic. So what is a rare disease? Well, it's a disease that causes harm, either it causes a chronic disease or causes mortality. Most of them have a genetic basis to them, probably about 80% are caused by aberrations in the genome, and these can be passed down from one generation to the next, depending on what the genetics look like. So, what is a rare disease? Well, most people agree that a rare disease is one where the incidence is less than about 1 in 2,000. So we're talking about things that actually occur infrequently, very infrequently. However, when you look at the number of rare diseases, there are roughly about 6,000 to 8,000 individual rare diseases. So when you add those all together, you get roughly 6 to 8% of the Australian population will be suffering from a rare disease. So individually they're very rare, but once you sum all of the people suffering from rare diseases, they become obviously more common. So rare diseases as a whole is a very important part of Australia's health, but individually, and each of these is different, it's very difficult to get researchers to actually work on many of these really rare individual diseases, and you will find that many of the families that actually have these diseases end up actually having to convince researchers to work on their own diseases. And they do this by actually getting together with people with same or similar diseases and forming groups of people that then try to convince researchers to work on these diseases by providing funds. So these are sort of individual disease-based philanthropic organisations, and they're very effective often at actually convincing people to work on the diseases. And when you see some of these diseases, which are often very debilitating, you can definitely understand why families and friends and everybody are interested in having people work on them. So, without further ado, I would just like to introduce the first of our 3 speakers, and this is Associate Professor Phillips, who's a clinician academic, worked for nearly 20 years with marginalised populations with special interest in the management of complex and multimorbid conditions. She has higher qualifications in anthropology and public health and is the chair of Social Foundations of Medicine at the ANU Medical School. That's that building that's just the other side of John Curtin School, Medical School. Very good. In her work as Medical Director of the ACT's Refugee Health Service and Chair of the National Network of Refugee Health Services, she is familiar with the challenges of caring for many patients with rare diseases and fragile social supports. So we're very fortunate here to talk to you tonight. Christine, thanks very much.
Do I start this by pressing on myself? Yes. Can you hear me up the back? Thank you very much for asking me to come and speak on this topic. When I was asked to come and speak, I thought, I'm a GP, I don't deal with rare diseases, which is a stupid thing to think because actually rare diseases account for about 6% of the patient work in general practice. So all of us see rare diseases, and at that very moment, as I said, I don't know that we see rare diseases, I was in my refugee health clinic looking at some information that had just come from a microassay about a patient of ours who was clearly had an undiagnosed rare disease. So and I sat and thought about it, and I thought actually in my population of patients who are newly immigrant populations, rare diseases are actually very, very common. And I want to speak to you today about the social consequences of having a rare disease in the context of having fairly fragile social and family supports. That's an image of a lone refugee looking across at Telstra Towers. That's an image of being rare and alone. So there's a statement in general practice that you should think, when you see a condition, you should not think zebras, you should think horses. You should think that common things occur commonly. But actually rare things occur commonly as well. And there are reasons why, particularly in a refugee health service, newly immigrant populations that the country hasn't had before, rare diseases may be even more common. We may be dealing with an unknown condition in a newly arrived community for which we do not have information. We may be dealing with a known condition in an unprepared community, and we may be dealing with also the consequences of war. It's been a condition of some concern in the refugee community internationally about the exposure to chemicals such as have been used in Syria and depleted uranium as used in Iraq, and the consequences of those for people. Now today I'm going to be speaking about an example of the first and the second category. These are really important issues because they relate to whether or not a visa will be issued in the very first place, whether they will be considered well enough to get a humanitarian visa and come to the country. Now, Australia increasingly does not exclude people from a humanitarian visa on the basis of their illness, but it did for many years. It's only been about the last 6 or 7 years that disabled people have been not have been reassured that perhaps they could come to the country. We're going to be talking about issues about diagnosis, issues about whether they can be supported for their disability, support for the family, and importantly, access to the social safety net. In particular, I'm going to be talking about the issues of what happens when you do not have a diagnosis. Let me give you this example of Mei Sun. That isn't Mei Sun, that's just a picture from the internet. But it is a Chin person, and Mei Sun is a Chin little boy, and that's from Myanmar. So he's a community from Myanmar who many of them have escaped and are living in camps along the Thai-Burma border. He came to Australia at the age of 3, having been born in this camp, and his initial health assessments conducted first of all by the International Organisation for Migration, and again here in Australia, all were fairly straightforward. It didn't look like there was any problem with this little child. He was a healthy kid. 2 years later, though, at the age of 5, became apparent that he had delays in speech. His mother reported he tended to fall over. He was friendly at school and angry at home, and he was referred back by this school for us to say, does he have a diagnosis of ADHD? And also a comment that he had obvious dysmorphic features, written by the school. He looked like that. So I couldn't really see that he had obvious dysmorphic features at all. But I've had a number of our patients from newly arrived communities who've been sent to us saying this person has an obvious dysmorphic feature. In fact, I've had Dinka patients who are patients from South Sudan, very tall people who've been sent to me with a provisional diagnosis of Marfan syndrome, whereas in fact what they have is just long arms and long legs. It's a normal feature for them. So I wasn't convinced that he had obvious dysmorphic features at all. The family didn't actually think he had a problem. The specialist services proposed that many of his symptoms could be related to trauma, and that brings up a cross-cutting feature that slows down diagnosis for our population, in that trauma is often very common as well. They've often lived very difficult lives. And so some of their behaviours and their responses and the learning delays may reflect trauma. So nobody had an actual diagnosis, and in the absence of a diagnosis, what actually happened was that his mother would now need to find work next year. So under the social safety net of Centrelink, she could no longer care for him after the age of 7. The school could not provide supports because nobody actually knew what was wrong with this child. He had uncertain access through the NDIS to the Early Childhood Early Intervention program. The mother couldn't access carer payment or carer pension, and there was no support to access public housing any quicker than anybody else, and public housing, if you're on a waiting list here, will take you at least 2 years at the most. I have patients who've been on the waiting list for public housing for 8 years. So there was nothing because he had no diagnosis for what was going wrong with him, there was no rationale for us to be able to advance his access to and his family's access to the social safety net. Over the next 3 years, he continued to struggle at school. A keen teacher organised review 2 years later. He was lost to us, really, and brought him back. And the mother had become concerned because she'd had a new baby, and that baby was actually meeting all her milestones. Mother and father at this stage were working as cleaners at night, so time-honoured new immigrant job in Canberra, cleaning your offices at night. So they were both doing that. Around about this point, the microarray was conducted and it showed a copy number variant. So we had something, but the significance of that was uncertain. So we're still in a situation where the child's prognosis and what we may expect of this child is uncertain, and the access to the social safety net remains patchy. So why does diagnosis matter? And I want to point out here that over the time I've been working in refugee health, proper diagnosis has become more and more important in terms of functioning as a barrier to whether people can access the social safety net. So that is public housing, Centrelink, even support for school, NDIS. If you want to get NDIS over the age of 7 years, you need a diagnosis. Disability support pension requires a diagnosis. Carers pension or payment requires a diagnosis. Any advocacy for public housing about increasing the priority or the types of housing requires a diagnosis. Now, you may think that access to public housing should have shelter, should have nothing to do with whether you actually have a diagnosed illness or not, but in the ACT it does. Funded teaching support requires an assessment of need and prognosis. So it is very important to this child that we can actually slot him into some kind of diagnostic category. And at the moment, we just have a kid who's a bit delayed, has some behavioural problems, is really not functioning at school, but doesn't meet any kind of diagnostic category. We can say maybe this child has a genetic disorder, but we don't know. It seems increasingly, though, this child does have a rare disorder, and we are really marking time as we wait by this child for the condition to start to declare itself in more physical ways, but we do not have a title of the disease. So for disadvantaged Australians, so this is the point I want to make, having a rare and undiagnosed disease means they have structural barriers to receiving the supports they need to cope with the disease. That is a sunk event of the way that we decide who is a good candidate for the social safety net. However, it's not just the case that if you know the diagnosis, you can access these. Here is a case of somebody for whom the diagnosis is abundantly clear, but her rare disease is so rare in this country that she has actually become ill across her entire lifespan. So this is Aisha. That isn't Aisha, that's just a picture from Salt magazine. The person that this case is based on comes from, in this case I'm saying she comes from Guinea. Now she came to Australia at the age of 5. She had a sickling crisis and diagnosis 2 days after arrival. So almost as soon as she arrived, she had acute pain in her abdomen, went to hospital. We thought she had malaria. Turned out she had undiagnosed sickle cell disease. You may think that's a bit odd that somebody should come over here and in the refugee without a diagnosis when as soon as she arrived here it became apparent, but in the refugee community it's not at all unusual. I think it's the very long flight from West Africa to Australia results in dehydration, so it makes people who have functioned, kids who have functioned all right previously, actually their condition declares itself on arrival. Throughout her life she has had repeated crises leading to 6 to 9 admissions a year. She takes her medication. She regularly goes and was taken by her mother to the haematology clinic. She's up to date with the immunisations needed for functional asplenia, which is what she's has, and she nearly dies of sepsis at the age of 14 years. So her journey in Canberra is that her sickling crises, which mainly involve joints, are triggered by extreme cold and extreme heat, both of which we have here. Every year I've had to write a letter to the school saying please let her start late, don't let her wait at the bus stop at 7:30 in the morning because you can predict what's going to happen to somebody with sickle cell disease. And every year the school would say, oh okay, we'll let her start a bit late. She's now graduated and at a technical college who will not let her start late. and so she's actually lost access to her education. Her mother can't work because of the need to care for this young woman now who's in and out of hospital all the time. NDIS does not recognise sickle cell disease as a disability, so she can't access NDIS. Fortunately, a social worker at Centrelink actually did assess her separately and decide that she had, and gave her access to disability support pension. So the next 13 years since her diagnosis have really left her in a situation now where at 18 she's undernourished, underweight, frequently in pain, fatigued and in and out of hospital. That would be her life at the moment. Beyond a doubt, I think her life expectancy has been shortened by our inability to manage her rare disease, a disease which is not rare in other countries. Her family are impoverished by her disease. She can't obtain the education she wants because of a lack of attention, lack of recognition of her disability by the educational institution, and she can't obtain the allied health support that she needs, nutrition, caseworkers, physio, because she can't access the NDIS. She relies on charity streams of money for her medications through our service from donations. Now, I think this is an appalling situation and not unusual for people with a disease that is rare, but not rare in other countries. 1% of babies in Guinea have sickle cell disease, very low incidence in Australia. We don't know the numbers, but the numbers are slowly increasing with migrant populations. But we have no policy or guidelines on sickle cell disease in contrast to Canada, in contrast to the US, and in contrast to the UK and many European countries. We are well and truly behind this, and this results in very inequitable access to health care. So the point that I've been making here, I think, is that there are two types of knowledge and two types of blindness, like these little kids looking through the wrong end of the binoculars. It's important that we recognise, and we are able to recognise, the rare disease when it occurs. Giving a name to an uncertain set of illnesses and symptoms is really, really important for families. It's also really important for them being able to access the social safety net. But societal knowledge also matters. We should be prepared to recognise the impacts of a rare disease when we know what they are and ensure access to the social safety net. Rationing access to services by diagnosis, which is currently what happens, is inevitably inequitable to people from disadvantaged backgrounds and to people with rare diseases. Thank you.
Christine, thank you very much for that. I was waiting for the optimistic story at the very end, but it never came. So, our next speaker is Professor Matthew Cook, who is Professor of Medicine at ANU, Director of Immunology at the Canberra Hospital, and Director of the Centre for Personalised Immunology, or CPI, which is an HMRC Centre of Research Excellence. He's a clinician scientist with more than 20 years' experience in investigating immune deficiency and autoimmune pathogenesis. Matthew, thank you very much.
Thanks very much, Simon, and thanks for the invitation to come and speak this afternoon. So you can see from my title slide, I'm going to just consider some of the biological aspects of why some diseases are common and some are rare. And if we're going to consider these different types of diseases, we first of all have to think about what a disease is. This is a somewhat contentious topic. I'm going to take a biological definition, which specifies that a human disease represents some sort of incompatibility between a person and their environment. The important point to draw from this definition is that there is a relationship between a person's makeup and the environment that they find themselves in, and potentially someone might move environments and that might precipitate a problem. Sometimes there may be a problem with a person's individual makeup that will lead to a disease developing almost in any environment. So an additional concept to consider is what we refer to as the phenotype. Phenotype is a set of observable traits. So we might consider the physical appearance. Christine was just referring to this. Clinician had considered whether that little boy was dysmorphic or not. So physical appearance is part of the phenotype, but so is structure of the internal organs, their pattern of development. And then we can drill down even more in the laboratory. We can look at the biochemistry within the cells of an individual, the way their organs operate. These are all parts of the phenotype. We all have a phenotype. But sometimes a component of that phenotype we define as being abnormal, as being a disease. Now the counterpart to the phenotype is the genotype. And we'll just so that everyone's on the same page here, just summarise a little bit of molecular biology about what the genotype consists of. So most cells in the human body contain nuclei, and within the nuclei are chromosomes. 22 pairs of so-called autosomes, and then we have the sex chromosomes, X and Y. So each cell in the body has this set of chromosomes, and then if we drill down onto the chromosomes more, the chromosomes are made up of strands of DNA. And the DNA are these pairs, complementary pairs of bases. There are 4 types of bases: A, C, T's and G's. Now, the complete set of A, C, T's and G's that go to make up each of the chromosomes, and then in turn each of the chromosomes that go to make up all 46 chromosomes, are referred to as the genome. So the genome is the composite of all of the genetic material in the nucleus. And this is quite a big thing. So if we take the if we look at the makeup of the total amount of genetic material in the nucleus, there's about 3 billion pairs of A, C, T's and G's. So these are distributed across the chromosomes, and then the sequence is divided up into identifiable elements that are called genes, and there's about 22,000 of them. So the genes are made up of segments of these A, C, T's and G's. But collectively, the genes only account for about slightly less than 2% of all of the bases. Right, so each chromosome is a subset of this. So chromosome 5, for example, is made up of about 900 genes. About 900 of the 22,000 genes are present there. Now the genes, we're trying to understand how the phenotype arises. So the genes are made up of A, C, T's and G's, and this is a template for making another set of molecules called RNA, which are also made up of bases, very similar to the bases in the DNA. But then this provides the template for building proteins. So each of those 22,000 genes, many of them, are responsible for encoding proteins, and the proteins are made up of building blocks of 20 amino acids. And so if you work it out, if you take 3 of the bases, then that gives you sufficient diversity with some redundancy to encode all 20 amino acids. So triplets of bases are responsible for encoding amino acids. And so the 2% of the DNA that's responsible for encoding the proteins is referred to as the exome. All right, so the phenotype is highly variable. We know this. We only have to look around the room and we can see that the phenotype is highly variable. We look a bit different from each other, and then if we drill down to those other components of the phenotype, there's variation there as well. Now this variation in the phenotype is made up of variation in the genotype and variation in the environment. And these two components contribute. So in clinical medicine, we spend a lot of time characterising the phenotype. We've got all sorts of methods for doing this. Clinical assessment, but then we can run all sorts of tests and scans, blood tests to characterise the phenotype and to define the extent of variation. We've also spent a lot of time characterising environmental variables, and some of those are listed here. And of course you'll be aware that there's been a lot of effort to see whether these environmental variables are associated with some of this phenotypic variation. But until relatively recently, it's been quite difficult to characterise the genotypic variation because of the magnitude of this challenge of characterising the 3 billion bases and understanding how this might relate to the phenotype. But now we can start to do this, and this has given us important insights, particularly into rare diseases. So genomic variation refers to changes within that complete genetic makeup, within those 3 billion bases. And there are errors that arise all the time, or changes that are introduced into the genome all the time. And of course this is the basis of natural variation that underpins evolution. So genotypic variation is built into biology, and this is the reason for different species. But not all genetic variation is advantageous. Not all leads to a drastic change in the makeup of an individual. Many are neutral, many variants don't make any difference whatsoever, but some are deleterious, and those deleterious variants are those that are associated with disease. Now, this has been understood at a theoretical level for a long time, but now we've got an empirical basis for much of what I'm telling you about, and that's come about because of progress in understanding the human genome. And you will recall that the human genome was sequenced. It took about 15 years and $30 billion, large international collaborative effort to arrive at the reference sequence for humans. And this was published at the beginning of this century. And then over the next decade or so, progress was made. And one of the first things that was noted was that once a few more individuals were sequenced, we've referred here when the big announcement came to the human genome, but in fact that was the human genome of just a handful of individuals. And so once a few more individuals were sequenced, it turned out that there was quite a bit of variation in the genomes from person to person. And this spurred on a larger scale project to sequence 1,000 individuals, and then we really started to get an understanding of the extent of genetic diversity across the human population. Now, at the same time, there have been technological developments which have meant that it's now feasible to sequence the human genome in a relatively short period of time quite cheaply. And so, as I mentioned, it cost $30 billion. That was starting from scratch, so it's not really a fair comparison, but it was still a massive undertaking to assemble the reference genome in the first place, and now we can sequence the human genome for a couple of $1,000s. Now we can sequence the entire genome. We can sequence just that 2% that's responsible for encoding the genes. That's called human exome sequencing. And here in Canberra, we've been doing this for some time at the Centre for Personalised Immunology, so about a decade now, and this has been a fundamental basis of one of our major research projects. And more recently, we've introduced a clinical service So this is now in actual clinical practice. So one of Christine's patients could be referred and we could sequence their exome for diagnostic purposes. So this is now in prime time. Now, we have a much better idea now about the spectrum of human genome variation, and it really is extraordinary. So 3 billion bases, and about 1 in every 1,000 bases varies from person to person. And most of those are probably neutral, but there is an enormous amount of this. But about 100 variants in any individual, we would guess, confer a loss of function in the protein that's being encoded. And there's this very high rate of novel variants. So if we sequence an individual, we find 50 to 100 variants that we haven't seen in anyone else, and now there have been hundreds of thousands of individuals sequenced around the world, and this number isn't coming down any lower. It's a fact of biology. Right, so let's get back to this question. That's a bit of background. What about this question of why are some diseases common and some diseases rare? And it relates a little bit to the history of humanity. So there are some genetic variants that are present in all of us. So these are genetic variants that arose before humans evolved. So the gene that's responsible for the last step in the metabolism of vitamin C is like this. None of us can make our own vitamin C. We have to eat it. Common, and then at the other extreme are mutations or genetic variants that are present that we've only identified in one individual. So as rare as can possibly be the case. So these are variants that are present in an individual and aren't even present in either of their parents. So it's a brand new genetic variation that's just arisen in that generation. And then slightly more common are those that have arisen within the past generation or two, or where we might have found more than one family. So these are the rare diseases, and they are arising from genetic variants that are extremely rare in the population. There are also a subset of genetic variants that are present very commonly in the population, and this is thought to be because they arose a long time ago, maybe 60 to 100,000 years ago, and shortly after that there was a big explosion in the human population, and so genetic variants that were already present were propagated into substantial proportions of the human population. And each of those variants has such a weak effect, a weak deleterious effect by itself, that there still hasn't been enough evolutionary time for those variants to disappear from the human population. And so the genetic variants underpinning common diseases have been present for a long time, and each of them exert a relatively small effect. The more recent variants that are responsible for rare diseases often have a much more significant deleterious effect. So rare diseases are often single gene disorders, so monogenic single gene disorders, and they've arisen from recent onset variants, and these variants arising as a result of this measurable error rate in DNA replication. And that's what determines why they're so rare. Complex diseases, or common diseases, things like asthma and osteoarthritis, arise from common variants, and probably are the composite outcome of many individual variants that are relatively common within the population. All right, now how do we get from genes to phenotypes? Well, I'm not going to go through every element of this diagram. Save that for the medical students. But the important point here is that genes encode proteins, and proteins are part of networks which regulate how a cell works. And this means that sometimes there are important identifiable pathways where the products of different genes cooperate in order to transmit a particular type of signal. So there can be a combined action from the products of many different sorts of genes to lead to a particular biochemical or cellular outcome. So if we start to think about how this might actually relate to phenotypes, how the genotype might actually connect to the phenotype, then we can think about components of those sorts of pathways in this sort of diagrammatic form, where a cellular phenotype or a biochemical outcome is the result of the combined action of many different proteins within a particular pathway. And some of those proteins might be absolutely central to that biochemical or cellular outcome and might be referred to as core genes. And some might be more peripheral. They might interact in some way with the core gene but not be absolutely responsible. And therefore, if there's a genetic defect in the gene that encodes the core gene, this could be catastrophic and the pathway might be disrupted altogether, and this could lead to a disease phenotype. Whereas if there is a genetic variant in one of these interacting partners, then the biological system might be sufficiently robust to withstand that change. Nevertheless, if a person is unlucky enough to have genetic variants in a number of these peripheral genes, then that could result in a similar outcome to a mutation in that core protein. Right, so rare diseases overall we think are due to single genes. They're predominantly found in younger individuals, whereas we think of common and complex diseases as being the disorders of later in life. At least this used to be the dogma, but now that we have these tools to sequence individuals, it's not quite so clear-cut. And part of this relates to what I was just explaining on the previous slide. The other thing to consider is that the gene-environment interactions, remember, which account for the variation in the phenotype, are also not absolutely clear-cut either. If we think about scurvy, as I said, this results from, of course we all think this results from vitamin C, but it's also the combination of a deficiency of vitamin C, but it's only so because of a universal defect in all of us, which means that we can't synthesize our own vitamin C. So is this an environmental or is it a genetic disorder? Conversely, phenylketonuria, which is screened for on the Guthrie card, is a rare disease. But if we manipulate the person's environment appropriately with the right sort of diet, then we can minimise the disease phenotype even in individuals who have that rare mutation. Now, my particular interest is in immune deficiency disorders, and we know that individuals who lack antibodies and therefore are at increased susceptibility to infections can arise as a result of single gene disorders, and some of these present in young children, and these disorders follow the pattern of a typical rare disorder, prevalence of about 1 in half a million to 1 in a million. So that's pretty clear cut, and these individuals present early in life with recurrent infections. But we now know that there are also versions of adult onset antibody deficiency. And contrary to the speculation in the pre-genomic era, not all of these are due to the combined effect of multiple common variants. There's also a subset of individuals in this category that also have single gene disorders leading to this phenotype. But the nature of those genetic variants is a little bit different, and this is probably because the reason that the onset is delayed is because the effect on the protein which is being encoded is not quite as catastrophic as it is with the other variants, and there's the opportunity for some of those peripheral genes to modify the phenotype in these cases. Alright, so this is our current sort of thinking about these problems, that we might have different sorts of disorders which are arising in as a result of biology. So we might have a phenotype like obesity, where if we look at the genomic variation in a large number of individuals, we possibly won't find any of these core abnormalities. But we're nevertheless able to modify this phenotype by modifying the environmental factors. On the other hand, other disorders are clearly arising as a result of genomic variation, sometimes apparently a single gene variant, and therefore there is a qualitative difference between these individuals and those who don't have this genetic variant. And in these cases, it's unlikely that any amount of environmental variation is going to eliminate the phenotype altogether. Nevertheless, modern medicine tells us that even with catastrophic genetic variation, environmental variation can have a significant impact. Okay, so what we're left with is, from studying rare diseases and conceptualising how they actually are different from common diseases, we can start to think about all diseases in mechanistic terms. And this is very important because it means that we can start to take different approaches to treatment. If we understand mechanisms, particularly if we can identify those core pathways that might lead to the disease phenotype, then we've identified the best and most appropriate target therapy. And we call these precision therapies. And once we've introduced those precision therapies, then they're usually, one, highly effective, and two, minimise the risk of side effects because of a lack of off-target effects. The other thing that to take away from this, though, is that this distinction between rare and common diseases is not always absolutely clear-cut, as I explained with the adult onset immune deficiency. And we're now seeing across many different sorts of diseases small subsets where a single gene disorder leads to a very similar phenotype to those which we would normally expect to be accounted for by the composite action of multiple common variants. And while this creates difficulties for diagnosis, it also is very important for the reasons that I've just outlined in giving us new insights into how to go about treating these disorders. So I'll stop there. Thank you.
Yeah, thanks very much, Matt. And if I could ask Siobhan Bourke, is our next speaker. Siobhan is a research fellow in health economics in the Department of Health Services Research and Policy in the School of Population Health here at ANU. Her research is mainly focused on health economic evaluation and orphan drug policy in the UK. She completed her PhD in health economics on the economics of orphan drugs at Bangor University, which I presume is in Maine.
Wales.
Wales, is it?
Yep.
Okay, it's the original Bangor.
Yep.
In 2018. Thanks.
Thank you. Today I'm not actually going to talk about my research because it's very UK focused. So instead I'm going to talk about the US Orphan Drug Act, which is the original Orphan Drug Act. So patient advocacy groups for rare diseases have been raising awareness and improving the lives of people with rare diseases and their family. The most successful campaign was providing patients with rare diseases access to treatments by campaigning for legislation for orphan drugs. And orphan drugs is the general term that is used to describe treatments for rare diseases. The Orphan Drug Act is a legislation that incentivises and encourages the development of treatments for rare diseases. It has had a major impact on increasing the numbers of treatments available for patients with rare diseases. So today I'm going to talk about the background of the Orphan Drug Act, the issues it faces today, and how patient advocacy could still have a major impact in providing treatment for rare diseases. So why did this problem come about? Well, in response to the thalidomide tragedy in the EU, and if you're not aware of the thalidomide tragedy, it was a drug that was prescribed for morning sickness during pregnancy. However, the safety profile of the drug wasn't established, and thousands of children ended up born with birth defects. The US Senate produced the Kiefer-Harris Amendment Act. US senators strengthened the FDA's control of experiments on humans and changed the way new drugs are approved and regulated. So pharmaceutical companies were now required to demonstrate safety and effectiveness of their drugs before approval via randomized controlled trials. This increased the cost to pharmaceutical companies to gain approval, and as an unintended consequence of this act, drug companies began to neglect and orphan diseases, and that were diseases that would provide limited or small profits in favour of treating a larger market. And this is where the term orphan drug originated. So very little was being done to study these diseases or develop treatments. Only 38 drugs in total were approved in the US specifically for orphan diseases before the Orphan Drug Act. And to put that into context, there is 7,000 rare diseases and around 25 to 30 million patients with rare diseases in the US. So there is a high level of unmet need. So there was an increased movement to promote research into rare diseases. And this led to patient organisations lobbying for treatments in rare diseases, and they formed an umbrella group, the National Organisation of Rare Diseases, or NORD. And it targeted politicians to focus attentions on this problem. The organisations lobbied Congress, and when they finally got a hearing to bring attention to the problem of orphan drugs, it was unsuccessful. The event was poorly attended, but they did have patient advocates there who spoke about their struggles of rare diseases. And this led to an article in the LA Times, which was spotted by a TV producer who was actually suffering from a rare disease. Does anybody recognise this person? No? Well, it's before my time as well. So the producer was actually the brother of Jack Klugman, who is also known as Quincy M.E. And for those of you who are not familiar with the programme, this is a precursor to CSI. So a medical examiner who used forensic medicine to solve crimes. So in the episode, they focused on the mysterious death of a patient with rare diseases. And while the patient wasn't murdered, the episode highlighted the neglect that rare diseases was experiencing in drug development. And then the rest of the episode really focused on the Orphan Drug Act. And this led to a new congressional hearing, which resulted in the Orphan Drug Act. The Orphan Drug Act has had a number of incentives for pharmaceutical companies to produce orphan drugs. So what did it include? Well, we have both financial and non-financial incentives. So for the financial incentives, it provided research grants for pharmaceutical companies in rare disease, to research rare diseases. It also gave them a tax credit, so they could claim 50% of their R&D costs back from the government. It gave them a monopoly, so marketing exclusivity of 7 years. And it had user fee waivers, and that could add up to about $2 million. It also had non-financial incentives. So this was fast-track approval. So through the FDA's designation review cycle, it was about 90 days, which is significantly faster than a non-orphan drug. It also provided free protocol assistance and any assistance in developing the consultation. And what was the impact? It has been highly successful. It's gone from 34 drugs to 730 on the market in 2018. Over the past 30 years, there's similar legislation has been created in Japan, here in Australia, and in the EU. While the availability of these drugs have been somewhat fixed by the Orphan Drug Act, problems still remain for patients of rare diseases. The monopolistic nature of orphan drugs allow pharmaceuticals to charge extremely high prices. And now we have, again, unintended consequences of an act, just like the Keever-Harris Amendment Act. So the Orphan Drug Act is being used by pharmaceutical companies' advantage in ways that its policymakers didn't foresee or intend. People believe now that the incentives are too lucrative for pharmaceutical companies and that there are too many loopholes in the Act. One way in which pharmaceutical companies are exploiting the legislation is by salamitizing, so creating artificial rare disease subsets. So genetic research in Parkinson's disease, which we thought was once just one disease, is actually a collection of at least 5 distinct illnesses, each caused by its own unique genetic mutation. They also can apply for multiple indications. So there's 80 orphan drugs approved by the FDA for more than one rare disease, and in some cases, multiple rare diseases. So Botox, for example, is an orphan drug, and it has 3 orphan drug indications. And this increases the number of patients being treated by this high-cost drug. Another way the Orphan Drug Act is being used is by sequencing orphan drug approval processes. So that's stacking up the series of orphan drug disease approvals. So one 7-year exclusivity leads on to another. So this maximises the length of the monopoly that the pharmaceutical company have. And we see this with Curtin. It's a metabolic drug disorder, or metabolic drug. For more than 20 years it has had a monopoly. It received its second orphan drug approval 4 months before its first one was about to expire. And it won its third orphan drug approval by slightly tweaking the formulation for an IV formulation of the drug just 1 day before its exclusivity period ended. Another common problem is the repurposing of old drugs gaining orphan drug status. So these drugs are entirely new or innovative. So for example, in the UK, ibuprofen, which costs less than 10p per gram, has an orphan drug indication. And this orphan drug counterpart costs 82,000 times more, despite it being the same drug. Possibly the greatest unintended consequence of the Orphan Drug Act is the cost of orphan drugs. The top 5 most expensive drugs in the world in 2018 were all orphan drugs, and the prices range from $365,000 to $1.2 million per year. The median cost per patient is 5.5 times higher for orphan drugs compared to non-orphan. Only 24% of orphan drugs cost less than £10,000 annually. 58% cost between £10,000 and £100,000, and 18% of orphan drugs cost greater than £100,000 annually. Pharmaceutical companies argue that to fund future research and to recoup the cost of developing these drugs, they must charge these prices. But this in turn has an effect on healthcare budgets, putting greater pressure on governments to provide these drugs. So while the Orphan Drugs Act has improved availability, it hasn't improved access. So the reimbursement of a drug generally will depend on its cost effectiveness. That is, its value for money for the healthcare system. Orphan drugs rarely meet these standards, but are often approved for reimbursement despite limited clinical and economic evidence. In Australia, the reimbursement recommendations are made by the Pharmaceutical Benefits Advisory Committee, PBAC, And despite their flexibility, they do regularly reject funding for orphan drugs. So to combat this, they've created the Life-Saving Drug Programme, and when it was established with 8 criteria for funding, so rare disease patients have access to expensive life-saving drugs. However, they're not actually considered for this program until they've been through the normal PBAC evaluation methods. So there is a delay in patients accessing these treatments. And how much does the Life-Saving Drug Programme cost? Well, they subsidised, in 2015, they subsidised 10 medicines for eligible patients at an average cost of £311,000 or $311,000 per patient annually. In 2014-2015, the programme treated 289 patients at a cost of approximately $85.9 million. However, the funding of these very expensive treatments may mean that funding of other services and other patients, including those with rare diseases, may have to go without certain elements of healthcare. And this is a challenge that is faced by most countries, to provide an equitable and fit-for-purpose healthcare system for all patients. So what is the future of the Orphan Drug Act? Well, pharmaceutical companies can charge exceptionally high prices for orphan drugs, so it is unclear whether or not there is a need to incentivise research into rare disease therapies. And we have seen in 2018 there is actually an erosion of support for the Orphan Drug Act. In the US, the Tax Cuts and Jobs Act reduced the tax credit for orphan drugs from 50% down to 25%. And we've seen that in other jurisdictions, with the EMA publishing guidance to try and reduce and close the loopholes. So what can we do? Well, the Orphan Drug Act was there for neglected diseases. So why don't we put the focus back on neglected diseases? So we should change the wording of the orphan drug policy so that we incentivise research into into diseases that have been ignored by major pharmaceutical companies, rather than this arbitrary number of 1 in 2,000 patients. So we should say it's a neglected disease that we are focusing on, not the total number of patients. So this would close the loophole and go back to what the Orphan Drug Act was intended to do. And I said as well at the beginning that there is still room for patients and patient advocacy. And this is outside of policy. It is where patient advocacy is central in gaining additional treatments for rare diseases. And Rare Disease Day is a celebration for this. So how do we look to the future, and how can patients be the driving force of change? Well, this is a suggestion by researchers in the UK who propose a greater role for non-for-profits, so that include patient advocacies and universities, to become market access holders, so they take the role of the pharmaceutical company. Looking at drug repurposing. So this is what the pharmaceutical company do. And if we apply that to existing drugs, and we use that for the treatment of previously unconsidered indications. So like ibuprofen, the ibuprofen situation I discussed earlier, a common disease drug is repurposed to treat a rare disease by having the benefits of the orphan drug policy and setting a fair price for treatments. And having this ownership of intellectual property rights by a socially motivated organisation may facilitate access to more affordable orphan drugs, and this for the benefit of patients and the healthcare system. And this model can have a significant contribution to bringing affordable orphan drugs to the market and creating a more equitable healthcare system. Thank you.